Protein purification. In this section, we are going to explain some purification methodologies that are carried out in our service. There are so many methods that can be applied to purify proteins. The chosen method depends on several factors such as the cell compartment where is the protein characteristics, the needs of the client in terms of purity, activity, etc. Cell lysis and solubilization. To purify proteins that are not secreted to the extracellular medium, it's necessary to lysate the cells to take out proteins from the intracellular compartment. There are several methods of cell lysis with different efficiency depending also of the tissue and the cell type. Gentle methods for cells that break easily, such as osmotic shock, freeze-thaw cycles, detergents, enzymatic digestion. Harsh mechanical lysis such as sonication, disruption, grinding, beats, etc. The first step is to take the pellet kept at -80 °C and thaw it in ice. The pellet is resuspended in the appropriate lysis buffer suitable for protein stability and compatible with later purification methods. It's very important to add to the lysis buffer protease inhibitors to avoid the digestion of the protein to be purified. In this case, we are adding some tablets from a commercial ETA-free protease inhibitors cocktail. The effect of these inhibitors only lasts few hours, so it's very important to keep the sample as much as possible refrigerated to diminish protease digestion. The pellet must be very well resuspended in this buffer, trying to dissolve all cell lumps in order to have a more homogeneous cell lysis. In this case, we use a disruptor as a cell lysis method. This is an homogenator that passes the cells at high pressure through a valve disrupting cell membranes. The sample is passed several times to be sure that a high percentage of cells are disrupted. Then the cell lysate is centrifuged during 45 minutes at 15,000 Gs to separate cell debris and insoluble proteins contained in the pellet from soluble proteins present in the supernatant. In this supernatant, a high amount of several soluble proteins is present, and it is necessary to proceed with protein purification to obtain the protein of interest as pure as possible. In some cases, the protein of interest is not soluble in the lysis buffer and accumulates in the insoluble fraction, the pellet. This phenomenon sometimes occurs when the overexpressed proteins are unable to be folded properly, aggregating in the form of inclusion bodies. When this happens, the first step is to try to optimize the protein purification to allow the cell machinery to adapt to the overexpression of a foreign protein. When this is not possible, then a solubilization of the insoluble proteins must be performed. For the solubilization of insoluble proteins, a variety of detergents can be used with different solubilization and denaturing capacity, being in some cases necessary to renature proteins later. Some ions can also help to solubilize them by preventing protein interactions that could produce aggregations. Adding reducing agents also helps by preventing hydrogen bonding. The pellet, composed by the insoluble fraction, is resuspended in a buffer with 0.5% of the N-luryl detergent. The sample is then incubated under agitation to allow the solubilization of the insoluble proteins. In the case of this protein, an incubation of 40 hours at room temperature is performed. Later, the sample must be centrifuged to separate insoluble components in the pellet from those solubilized in the supernatant. This supernatant will contain several solubilized proteins, so it's necessary to proceed with protein purification to obtain the protein of interest as pure as possible. Now we move to protein purification methods. Protein purification methods. Here there is a list of the most common protein purification techniques that are used to purify proteins. Affinity chromatography is based in the affinity of the protein of interest to specific components such as metals or antibodies, where the protein binds with a high specificity. Ion exchange chromatography is based in the affinity of proteins to an ion exchanger. Positive or negative charged proteins interact electrostatically with an immobilized opposite charge. Hydrophobic interaction chromatography separates proteins according to differences in their surface hydrophobicity. Size exclusion chromatography, also known as gel filtration chromatography, separates proteins by their size. All these techniques are carried out by using specific resins normally packaged in columns to be used in FPLC systems, fast protein liquid chromatography. But there are other protein purification techniques that can be performed: selective precipitation, centrifugation, immunoprecipitation, etc. In this section, we are going to show the IMAC and SEC methods. Protein purification by IMAC, immobilized metal affinity chromatography. IMAC is the most widely used method to purify proteins according to their affinity to specific metal ions such as cobalt or nickel. Here we show a scheme of an IMAC column. In yellow, resin bits with attached metals bind proteins through their histidine tags, whereas the rest of the proteins passes through the column. The specific binding is reversible and the proteins are eluted later on by using specific elution buffers. Before starting the purification procedure, it's necessary to prepare the sample. First, it must be filtered through 0.22 micrometers filters in order to remove cell debris that could clog the resin columns. Later on, the pH must be checked to be sure that it's above 7, since the pK of the side change of histidines is 6.0. This is an essential step to assure that histidine tags bind to the immobilized metals. For protein purifications, we use columns connected to a FPLC system, fast protein liquid chromatography. This equipment is composed of several parts, a main unit where there are located the chosen column, the pumps, the ultraviolet pH and conductivity detectors, the injection loop, valves, etc. For large volume samples, there is an external pump to load the sample into the column. The fraction collector is a rotating rack that automatically collects fixed volume samples in separated containers. And finally, a computer with a specific software to record all data and to control manually and automatically the FPLC operation. The first step is to connect the chosen column to the system. In this case, a cobalt column is connected to the column valve. It's very important to be careful connecting the tubes in order to avoid the entrance of air in the system that could disturb the liquid flow through the resin. Inside the whole system and columns, there is a solution of 20% ethanol in order to prevent the growth of microorganisms. So then, the first step is to wash them with ultra-pure water and purge the pumps to avoid the entrance of air in the system. Once the ethanol is removed, a subsequent wash with binding buffer is performed in order to equilibrate the resin in the same buffer of the sample. Once the column is equilibrated, the sample must be injected into the column. For this purpose, first it's necessary to load the sample inside the injection system. Depending on the volume of the sample, a loop for some microliters or a superloop for a maximum of 50 milliliters can be used. For large volumes, the superloop can be loaded several times or directly use the external pump. The sample must be loaded in the injection system, ensuring again that no air enters. The injection of the sample to the column starts. From this moment, all the process is automated through a programmed method. The plunger descends to inject the whole sample into the column. Proteins with affinity bind to the resin, whereas the rest of proteins pass through the column and are detected by their absorbance in the ultraviolet range. The sample that doesn't bind to the resin, called flow-through, is collected in a tube in order to be able to recover the protein of interest in case it hasn't bind to the column. Here in the chromatogram we can see in blue an increase of the ultraviolet absorbance coming from unspecific proteins that don't bind to the resin. This absorbance is maintained stable during the whole sample injection. At the end of the injection, a brownish coloration of the column can be seen, indicating the presence of proteins. However, these proteins have different binding specificity to the resin, and it is necessary to proceed with a column wash to remove non-specific proteins weakly bound to the column. The washing is stopped when the chromatogram shows a decreasing of the ultraviolet absorbance to near the baseline, indicating that no more proteins are removed from the column. The wash is also collected in order to be able to recover the protein of interest in case of being removed during the washing step. It's time for the elution of the proteins bound to the resin. To this purpose, a gradient of the elution buffer is done. Its concentration is gradually increased to be able to separate proteins with low affinity from those with high affinity. As an elution method, we normally use imidazole, a histidine analog that competitively elutes the bound polyhistidine residues by disrupting the binding between histidines and metal. The eluted proteins, previously detected by the ultraviolet cell, are collected in tubes in a fixed volume. Each tube collected is called fraction. During fractionation of the eluted proteins, the chromatogram shows, in red numbers, which fraction is being collected in every moment, being able in this way to perfectly correlate each fraction with the ultraviolet absorbance. Fraction containers are numbered with the fraction number and kept in ice for later handling. During the elution, a rapid increase in the ultraviolet absorbance indicates a massive elution of proteins. At this point, the gradient must be stopped and maintain the same concentration of the elution buffer in order to avoid the overlapping of different eluted protein populations. The ultraviolet absorbance shows a peak profile that can be correlated with the fractions collected, clearly seen in this zoomed view. Once the absorbance decreases because no more proteins are being eluted, the gradient can be continued to obtain more other peaks. However, in this case, because of previous experience with this protein, we already knew that only one more population were going to be eluted. So then, in order to have a more concentrated sample, we decided to stop the gradient and elute the protein directly in 100% of the imidazole solution. Few milliliters later, a second peak is obtained, where it is supposed to be the protein of interest. At the end of the elution, the resin recovers the pinkish coloration, indicating that all proteins have been eluted from the column. Now it's time to check by gel electrophoresis and Western blotting that the protein of interest is in the second peak. According to the chromatogram information, some fractions of each peak and the collected flow-through and wash are analyzed. We use specific polyacrylamide gels with the TGX technology that allows to visualize protein bands by ultraviolet excitation of aromatic residues, avoiding in this way the coma-C staining. The visualization of the gel shows the presence of multiple bands in the initial sample and the flow-through, but also in fraction of the first peak, indicating that a variety of unspecific proteins were eluted in this peak. However, in the second peak, a band around 25 kilodaltons appears compatible with the protein of interest. To be sure that this band corresponds with our protein, it's necessary to perform a Western blotting using antibodies against the histidine tag. To this purpose, proteins are transferred from the polyacrylamide gel to a PVDF membrane using a semi-dry transfer system. The chemiluminescent reaction shows that effectively the band of 25 kilodaltons from the second peak corresponds to a histidine-tagged protein. So then, in the first peak of the chromatogram, there is a wide variety of unspecific proteins without histidine tag, whereas the second peak has a quite pure protein whose molecular weight and the presence of a histidine tag suggest that this is our protein of interest. All fractions from the second peak are pooled, and depending on the interest of the client, the pool can be prepared to be delivered or to carry out the next purification step. In any case, the protein concentration of the sample is determined by measuring the absorbance at 280 nanometers and taking into account the molar extinction coefficient of the protein. Before the delivery of the protein or next purification step, the protein pool must be dialyzed in order to remove the imidazole and to contain the protein in the buffer of the customer's choice. To this purpose, the sample is introduced in semi-permeable bags that are submerged in a high volume of the desired final buffer. To promote buffer exchange, the system is kept refrigerated under agitation during several hours, normally overnight, and the buffer is renewed several times. Once the dialysis has finished, we check the presence of protein aggregates. Here, in this picture, it can be seen several degrees of protein precipitation. This phenomenon occurs sometimes after the dialysis when the chosen buffer isn't compatible with the purified protein. In these cases, the protein can undergo structural changes exhibiting hydrophobic regions that will promote its aggregation. When no information is available about the compatible buffer, it's necessary to carry out a macrodialysis analysis with several buffers to look for the best one. The macrodialysis technique will be explained in more detail in the protein characterization section. The sample, which is now in the buffer request by the customer, is taken out of the dialysis bag. Just in case, the sample is centrifuged during several minutes to pellet all the aggregates that may have formed during the dialysis. The protein concentration is determined again to know the protein lost during the dialysis. At this moment, the sample can be delivered to the customer or continue with the next purification step. In both cases, it's usually necessary to perform a concentration of the sample, that is carried out by centrifuging the sample in tubes with a filter that allows the passage of the buffer but not the protein. In this way, the sample is delivered at the concentration required by the customer, or in case of continuing with the purification, the sample volume has been reduced to perform the next step, the preparative size exclusion chromatography. Preparative size exclusion chromatography. This method is normally carried out as an additional purification step when extra purity of the protein is needed. Size exclusion chromatography is a method in which proteins are separated by their size. The region of the columns is composed by small porous bits. Proteins smaller than the pore diameter will enter inside the channels of the bits, whereas larger ones will flow outside them. So then, larger proteins flow through the column faster than the smaller ones since they have a longer way passing inside the bits. There are several column options with different resin types, with a variety of pore diameters of the bits, with different column volumes and resolution of protein separation. In this case, we are using the column high-low 16600 SuperDex that gives high resolution and allows a high volume sample loading. This is a column with 120 ml dextrans polyacrylamide bed that allows to separate proteins between 10 and 600 kilodaltons. Once the ethanol has been removed from the system and the column has been equilibrated with the running buffer, which is the same that the dialysis one, the concentrated sample must be loaded to the injection system. It's very important again to avoid the entrance of air in the column. In this particular column, the maximum sample volume allowed is 5 milliliters. It's important not to exceed this volume to avoid an excessive dilution of the obtained samples. Once the sample is injected on the top of the column, the running buffer passes through the resin in a fixed flow, moving in this way the proteins to the bottom. Proteins coming out of the column are detected by the ultraviolet cell and are fractionated in a fixed volume in the collector. The software shows in blue the appearance of a main protein population. The obtained chromatogram shows three peaks. The first one corresponds with aggregation of the protein of interest that is in the second peak. The third peak is composed by other smaller proteins. The narrow symmetrical shape of the second peak will demonstrate that the sample is structurally homogeneous. Fractions from the second peak are pulled and concentrated if necessary. By using size exclusion chromatography, a more pure and homogeneous protein has been obtained, eliminating aggregates and unspecific proteins of different molecular weights that were purified with our protein in the IMAC method. Protein purification with magnetic beads. In the last part of this section, we are going to explain a protein purification method based on the protein affinity to functionalize magnetic beads such as dynabeads. Dynabeads are 1 micrometer diameter spherical magnetic particles that react to magnetic fields. The beads are composed of an iron core surrounded by a polymer layer that can be functionalized with several affinity components, such as antibodies, ligands, metals, etc. Here we are going to show the process of protein purification by using cobalt functionalized beads to bind histidine-tagged proteins. In short, it's a IMAC method in suspension. This method is very fast, easy and cheap, and it takes about half an hour. It's appropriate to be used to set up the IMAC technique, being able to carry out several purifications at the same time to look for the best buffers. It can also be used to check that the IMAC method works with a particular protein before starting a complex IMAC purification by FPLC. This method can be used to purify big structures that could clog the IMAC columns, such as VLPs or bolts. However, the binding capacity of dynabeads is much less than that of packaged column resins, but it can be used as a final purification method when small amounts of protein is needed. The magnet used in this case is this 6 Eppendorf rack with round magnets located on both sides, but other magnet models for larger volumes and large number of samples are available. Dynabeads with this brownish coloration are stored in a conservative buffer that must be removed. So then the first step is to take the desired volume of beads and place them inside a tube and introduce it into the rack. Dynabeads are attracted by the magnet. 2 minutes must be waited to ensure that all beads have bound. At that moment, it's possible to aspirate the buffer and to resuspend the beads first in ultrapure water to clean them, and later with the binding buffer, repeating the process of separating the beads with the magnet. Now is the moment to incubate the beads with the sample. The beads are suspended in this case with a sample containing the green protein GFP with a histidine tag. The tube is then maintained under agitation during 5 to 10 minutes in a roller. We have chosen the GFP protein in order to better show the purification method. Once the incubation has finished, the tube is placed into the magnetic rack to separate the beads and remove the unbound protein and cell debris. This is the flow-through. Observe that the remaining sample has lost the green color, suggesting that the GFP protein has bound to the beads. Several washing steps are performed to remove unspecific bound proteins to the beads. In this case, some greenish coloration of the sample can be seen, indicating that some GFP proteins were weakly attached. Now is the moment to carry out the elution of the specifically bound protein. The elution buffer is added to the beads and incubated under agitation during 5 minutes. Pay attention to the greenish coloration of the eluted sample. Here we can compare the color of the samples obtained in each step. From the left to the right, it can be seen a high GFP concentration in the initial sample. The flow-through in the second tube with unspecific proteins shows an almost absent GFP. The third tube shows some amount of unbound GFP produced during the washing. And finally, the last tube with the eluted sample with high amounts of the green protein. With this last example of protein purification method, we move to the last section, protein characterization.